We will begin to discuss important concepts, techniques, and materials needed to cultivate microorganisms in the lab setting. In this video, we will explore important terminology pertaining to culturing microorganisms, concepts in clinical sampling, and procedures utilized to obtain pure cultures. One of the most important ways to identify microbes is to observe their growth on nutrients prepared in the lab. The cultivation of microorganisms is known as culturing. To culture microorganisms, an inoculum, or sample of the microorganism, is placed on a culture medium containing all the necessary nutrients required by the organism. The growth that results is known as a culture. Cultures may be grown in liquid media or on solid media. When growing microorganisms in a liquid media, or broth, growth is observed by cloudiness within the media. Therefore, one would not be able to determine if more than one species is present. However, when a solid media is used, growth is observed in a form known as colonies on the surface of the media. Colonies have distinct appearances when growing on the surface of solid media. As seen in the figure, colonies can differ in their shape, margin, elevation, size, texture, appearance, pigmentation, and their optical properties. These characteristics can aid in the identification of the organism. Microbial specimens can be taken from a variety of sources. Environmental microbiologists would take specimens from sources such as the soil, bodies of water, or the air. Clinical specimens, which are of special interest to this class because of our focus on medical microbiology, are samples taken from a patient that are to be examined for the presence and identification of microorganisms. The isolation and identification of pathogens is an important process in diagnosing and treating patients. Proper specimen collection is a crucial first step on which the isolation and accurate identification of pathogens are dependent. Healthcare professionals are responsible for the proper collection and transport of clinical samples. When a healthcare professional is collecting a specimen for microbiologic examination, a number of factors should be taken into consideration. The individual should collect the sample from the correct site without contaminating the specimen with microbes from nearby areas. They should use the proper equipment and transport media necessary for the specimen. They should take proper precautions to prevent spreading the pathogen to themselves or other individuals. And the specimen should be properly labeled and promptly delivered to the lab. The specimens are placed in media known as transport media that has salts and buffers necessary to maintain them for a short period and maintain their relative ratios. Samples that are not promptly delivered place the microorganism to be identified at risk of dying. As seen in the table, clinical specimens can be taken from a number of locations on the human body using a variety of methods. Bacteria rarely exist as a single species in the natural environment. However, properly studying a species requires that it be isolated from other species to obtain a pure culture. Pure cultures, also referred to as accident cultures, are cultures that contain a single species. A number of methods can be utilized to obtain a pure culture. The goal in any of these methods is to successfully separate one cell from a mixed population atop or within a solid media. This separated cell will give rise to a single colony. Each colony is then considered a pure culture. A colony-forming unit is the progenitor or original cell that gave rise to the colony. Microorganisms are not visible with the naked eye and are ubiquitous, meaning that they are literally everywhere. These characteristics make the study of microorganisms very different from studying any other living organism. Microbiologists must use aseptic techniques when studying microorganisms to exclude unwanted microbes. A number of techniques are available to isolate a pure culture. We will explore two isolation techniques, the streak plate and pour plate techniques. The streak plate is the most commonly used technique and one of the most economical in terms of materials and time required. A single loopful of culture is streaked over the surface of a solid medium in a petri dish. There are several ways to do this. The quadrant method of streaking is shown in the figure. In this method, a single loopful of bacteria is placed in the first quadrant and is spread 4 or 5 times across the first quadrant. The loop is then flamed and allowed to cool. The loop is then used to make 5 or 6 streaks from quadrant 1 into quadrant 2. The loop is again flamed and the same procedure carried out between the second and third quadrants and between the third and fourth quadrants. The plate is then incubated for the appropriate period of time to allow growth. This technique allows a concentrated sample to be diluted over the surface. Toward the end of the pattern, the resulting colonies should be separate from one another and may then be used to start pure cultures. The pour plate technique is also known as the loop dilution technique. This technique utilizes test tubes of medium containing melted agar that has been allowed to cool enough that it will not kill the bacteria, but not enough to allow the agar to solidify. A temperature between 45 degrees Celsius and 50 degrees Celsius. Inoculation of these tubes can be performed in various ways. Microbiologists may elect to use an inoculating loop and transfer a loopful of inoculum, or they may use sterile pipettes and transfer a known volume of inoculum. In the figure shown, a 1 ml volume of the initial sample is transferred to one of the fresh tubes of medium. This is then mixed and a 1 ml volume taken and added to another test tube of medium to further dilute the sample. This is typically done until 3 test tubes have been inoculated. The tubes are each then poured into petri plates. The agar is allowed to solidify, and then the inoculated plates are placed in an incubator to allow growth. Growth in petri plates following the pour plate technique is notably different from the streak plate. Because the bacteria are added to the medium and are mixed throughout it before pouring it in the plate, some bacterial cells may be found growing within the media and not on the surface alone. This technique can be useful in growing microaerophiles.